Good afternoon. So, welcome to the inaugural lecture, public lecture for the National Institute for Theory in Math and Biology. And before we introduce our distinguished speaker for this first public lecture, for those of you who don't know about our institute, I just want to fill you in a little bit about it. So the National Institute for Theory in Math and Biology was founded in 2023 as a joint partnership between Northwestern University and the University of Chicago. And it's jointly funded by the National Science Foundation and the Simons Foundation. So it's a public-private partnership in terms of forming a national nexus to better integrate the disciplines of mathematics and biology. That's our mission. And to fulfill this mission, we have faculty members and students and postdocs from both universities as well as other universities. We have a wide variety of convening programs that occur in the Institute space, which is not far from here. It's located on the 35th floor of the Hancock Center, or whatever the name of that building now is. Thank you. So the convening programs include workshops, multi-day workshops, one-day workshops, a summer conference, which will be held this August, as well as other impromptu seminars and so forth. And also it is a place where research is done, research in mathematical biology, as well as mathematics that's inspired by biology. So if you want to learn more about us, you can go to our website, NITMB.org. That's NITMB.org. And so to introduce our speaker today, Bill Kath is going to do the honors.
Oh, you should save it for Steve. So I was instructed to be short, which is very difficult because there are many, many things that I could say about Steve. He is the Susan and Barton Winokur Distinguished Professor for the Public Understanding of Science and Mathematics. And I don't know if that special professorship was created for Steve, but I would be willing to bet that it was, because definitely Steve epitomizes all of that description. But so those of us that work in academia, so the two major things that we spend our time on are teaching and research. And, you know, some people are good with teaching, some people are good with research, some are good with both. But Steve hits the trifecta. So he's has an incredible reputation for research, for teaching, and for public communication. And he's won awards in all three areas. So to give you just some ideas about the research side of things, I can't go into all of it, but his 1998 paper on small world networks with Duncan Watts, one of his students at the time, has been cited more than 50,000 times. So it's the academic equivalent of liking a video on TikTok, except in order to give that kind of a like, you can't just click on it. You have to write your own paper and cite it. So for people that look at citations, that is a huge number. So I mentioned he's awards for teaching as well. I won't go through those. In terms of public engagement with science, he has an award from the American Association for the Advancement of Science. And other winners are people like Neil deGrasse Tyson. He has the Lewis Thomas Prize for writing about science. And one of the co-winners, another winner of that award is Oliver Sacks. And for those of us that read the New York Times, many of you may have seen Steve's columns in the New York Times about mathematics, which was not the usual thing you read about in the New York Times. But I enjoyed all of them, and I'm happy to welcome Steve to talk about from math to bio and back.
Thanks a lot, Bill. That's a really very affectionate introduction. I appreciate it so much. And I'm really happy to be here with you for this inaugural lecture at NITMB. I've got the letters down. And thanks everybody for turning out. It's a nice, exciting crowd, very encouraging. Well, so speaking of the New York Times, I do have some new columns coming out in a few weeks. So if you do still read that newspaper, take a look. It's something really different. You know how people read on their phones nowadays with scrolling down like this? The columns are designed to be for that reader. So there are a bunch of images that are taken by a very artsy photographer of real objects, and then little captions of text followed by more images. So I hope, say, like a month from now, you'll take a look. They're very strange little, another attempt, like a new genre for math communication. I hope you'll like them. Okay, but anyway, back to our topic here for today. I want to give you 3 case studies from my own life that I hope will have more universal interest for you about this question of when we do mathematical biology, what should we be trying to do? And so that's why I'm saying like reflections on a two-way street. You know, on the one hand, we know that math can be really helpful in biology for mathematical modeling, for statistical analysis, for helping sharpen hypothesis testing. So we like to think that math has a role to play in biology. And obviously nowadays with computational biology, everyone knows that. So I was kind of brought up that the goal in mathematical biology is biology. That to the extent that the mathematician can help the biologist either in their experiments or interpretation, then you're doing honest to God mathematical biology. But there is another way of thinking about this, which is that biology is very inspirational in terms of the fantastic phenomena and kind of unbelievable things that happen in the biological world. Literally, like if someone told you about certain things, like if I told you there's something called a cell, and inside there are all these little machines that move around, and they're made of chemistry, and here's what they can do: tie knots in DNA and untie. I mean, it's like, really? Is that really possibly true? But except that, yes, it is. So biology, to a mathematician, is a fantastic source of new problems, new mysteries, and sometimes new mathematics comes out of it. And as a mathematician, that is very appealing to me. But it runs counter to what I was taught about what I'm supposed to be doing as a mathematical biologist. Like, is that cheating to see biology as a playground for mathematicians? Anyway, I want you to reflect on that, and I'm going to show you that in the course of my own life, and maybe for those of you that practice this, you'll think about this in your own career. Where do you fall on that spectrum? Do you have a dogmatic view of what the right way is to do theoretical and mathematical biology? Or do you take a more Catholic approach where anything goes? All right, so I'm going to show you, like I say, 3 case studies of my own attempt to do this kind of thing. So the first is actually from my first research of my life. I was an undergraduate at Princeton. Wasn't the easiest thing. I came in as intending to be a math major. I took the whiz kid linear algebra course, you know, for the people who did well, got a 5 on their BC calculus exam. And I got crushed in that linear algebra course. I was lucky to get the B- that I got. I really could have failed. It would have been a legitimate F. In terms of what I understood, very little. It was a super rigorous course all about proofs. It was supposed to teach us how to do proofs, and it didn't particularly teach me how to do proofs. I also understood, and I think it's been helpful to me as a teacher, what it means to feel so scared of an upcoming test, you know, that like you have to go to the bathroom from fear of what's going to happen to you. Until that time in my life, I used to feel like, bring it on. I can't wait for this test. I can show what I know. Not in that course. I didn't feel like that at all. In fact, I felt like I'm going to show you what I don't know. So, so that was the beginning of my math career at Princeton, a really unwelcoming start. And I kind of, the reason I'm telling you that is I didn't know what to do with myself. If I'm not doing math, what should I do? So I liked physics. I thought for a while I'd be a physics major. Then I had a really good complex analysis course in my sophomore year that was a math course. And I thought, okay, maybe I can do math, because that was just like calculus, but with complex numbers. And so I was back and forth with this kind of thing. And then my brother, he's passed away now, but my brother was 12 years older, and he was a lawyer. And he said one day when we were going home, I think for Thanksgiving vacation, maybe, I don't remember, but Steven, what are you going to do with math? Like, why don't you become a doctor? You know, and my mother had always said to me, why don't you become a doctor? You have very nice hands. That was, my parents had not gone to college. I do have longish fingers. So she thought, yeah, doctor. And after all, a Jewish family, you've got the lawyer, you should have a doctor, right? Other cultures, same thing. So I had no interest in biology. In fact, I took a biology course in high school where in the school that I went, they had this funny idea that chemistry builds on physics. So you should take physics first if you have enough math. So I had this weird education in high school where I took a physics-y first course in science, followed by chemistry, followed by biology, which you can see logically kind of makes sense, but it's not traditional to do it that way. And by the time I got to the biology course, I really wanted to learn special relativity and, you know, like more physics. And the biology course was so dreadful between meiosis versus mitosis and like, oh, please let me out of here. And I asked the teacher, could I just not take this course? And amazingly, she said, okay, but you have to promise to take it in college. It will be much more interesting. And do you promise to take it then? I said, yes, I promise. Not planning to really take it. But then, no, fast forward to the conversation with my brother, and he says, why don't you take all the pre-med courses? You're now a junior, and if you keep delaying this, it's going to get complicated. Like you'll graduate, it'll be much harder to become a doctor later. So why don't you just take all the pre-med requirements, and it'll be much easier, and you don't have to commit to being a doctor. You might actually like the subjects, might find them interesting. So I was a kind of dutiful brother, and I thought it was a good argument because I did generally like my science courses, although not that one biology course in high school. So anyway, I'm telling you all this because this is how I came to do mathematical biology. That conversation got me to, in my junior year, think about this, freshman biology, freshman chemistry, and organic chemistry at the same time. 3 labs a week, and I'm not good in the lab, and I don't cook. So that, because my teaching assistants would say, what is taking so long? This is just like cooking. And I would think, I don't even know how to boil water. You know, so like turning on the Bunsen burner is not that easy for me. Anyway, so I was pretty bad in the lab, but I really liked the biology course as I learned stuff about like how like what base pairing tells you in DNA, how the structure of DNA, the double helical structure in this amazing base pairing rule really kind of gives you a big clue about how DNA is going to replicate. And so I started to feel like there's geometry in this. And so in the second semester of biology, I had a great professor named John Bonner. If you know developmental biology, you will have heard the name John Tyler Bonner for his work on slime mold and other things. Anyway, Bonner was a fantastic professor who even at the end of the course brought in some scaling laws about metabolism of organisms of different mass. And there was real math in biology that I had no idea was there. So it wasn't just DNA geometry. There were all kinds of things that a mathematician potentially could do in biology. And I came to understand that by the end of my junior year. Anyway, so senior year, Princeton requires you to come up with a senior thesis. And I was a math major at that point. And I couldn't think, what would I do? but I asked my advisor, there was no applied math at Princeton at that time, but there was one person who had worked on soap bubble geometry, minimal surfaces. And he seemed like the closest thing to an applied mathematician, Fred Almgren. And I asked him, can you think of something that would combine geometry and biology? And he said, yes, you could think about DNA and what happens when DNA gets twisted so much that it's, instead of being a straight helix, it starts to become a superhelix. I hear that there's interesting problems about supercoiled DNA. So that was how I got started thinking about that. And so my first topic is about that. Chromatin structure. So at the time, there was a mathematician named Pohl, P-O-H-L, William Pohl in Minnesota, who said DNA is not really a double helix. Or not the double helix everyone thinks it is. This was 1979. And Pohl had this idea that, roughly speaking like this, that in, say, a bacterium, there might be 500 turns of DNA in the closed circular DNA. You know, DNA often doesn't have free ends. It can be formed into a loop, and 10 base pairs per turn, and maybe you have 5,000 base pairs. There'd be 500 windings of one strand around the other, and Pohl thought, how are those strands going to disentangle when DNA needs to replicate or segregate to different parts of a cell after cell division? And he argued that, okay, the biologists always said there must be enzymes that take care of this, but topoisomerases hadn't been discovered yet. So Pohl, a mathematician, said, I don't know if there are these enzymes, but I don't see how any enzyme could solve the problem, because the linking number of DNA, meaning the fact that one strand, like this would be a linking number of 1 if you do this with your fingers, like a single link in a chain. But with DNA, one strand in a loop, the other strand in bacterial circular DNA would be wound 500 times through there. And that's a linking number of 500. Now, how's an enzyme, which can only sense local things, it's much smaller than the whole global structure of the DNA, how can a little enzyme get the global information of what to do to that DNA? Like, even if it cuts it and then reseals it, which is what we now know happens, how do you know it won't make it linked 501 times? Like, how does it know which direction to go? Anyway, so this was in the air in 1979. How do you, what's the real evidence that DNA has this right-handed helical structure that Watson and Crick said? What's the really convincing evidence? And believe it or not, it was a live question. People were arguing, including Crick was involved in the discussion at that time. So I started thinking that's a good thesis topic, and I started learning about that stuff. And a friend of mine in one of my courses, a biochemistry course, said, we have a real expert at Princeton on DNA supercoiling. You ought to go talk to this guy. Okay, so that's how I got my first taste of interdisciplinary work. Okay, let me first remind you what chromatin is, because this is going to be relevant. Well, the person I went to talk to was going to tell me something about chromatin structure. And I'll get to the question in a second. But first, just as a reminder, so I've been talking about the double helix. There we are. Now it's shown here as having open ends, but don't worry about that. So there's the double helix. And then in the mid-1970s, people discovered these structures called nucleosomes. These beads, you could think of them as little spools, and the DNA winds around a nucleosome a very specific number of times. Like, you know, if you want to store a long piece of thread, you would wrap it on a spool. That's a nice organized thing to do. So nature has discovered that, except that what it does is it wraps DNA 1 3/4 turns on this protein spool, the nucleosome. Now, 1 3/4 is a weird number. So here's what I mean. Watch my hand do it. The DNA is going to come in. It's going to wrap around 1 full turn. That was 1. Then it's going to go 3/4 of a turn and come out at right angles to the way it went in. That was known from X-ray crystallography of these beads of nucleosomes at this time in 1979. So what you're seeing in this textbook diagram is the DNA is coming in, it's wrapping around 1 3/4 turns, and notice it's going out at a right angle compared to how it came in. Same thing with this one, the 1 3/4 turns on that, and so on. So this structure of beads on a string, as it came to be called, was known in the mid and late 70s when I was working. And there was an interesting experiment done at the time with tiny virus, SV40, simian virus 40, little mini chromosome, which was known to have 24 of these beads, and the DNA wound around those beads. And you could do an experiment where you knock off the nucleosomes and now look at the DNA, naked, so to speak, not attached to these protein nucleosome, the histones. And the interesting thing that was observed is that even though the DNA winds 1 3/4 times on each nucleosome, after you take the nucleosomes off, the winding causes this quantity, the linking number that I was describing, to be 24. With 24 nucleosomes, you get a change in the linking number of 24, as if each nucleosome was changing the linking number by 1, which seemed weird considering that you wound around 1 3/4 times, not 1 time. So there was a real question in biology: why isn't the linking number of DNA on the SV40 chromosome? Why isn't it 1.75 times 24? Why is it 1 times 24? And I didn't know that question, but that's what I learned from this biologist that I went talking to. Anyway, he was not interested in that question exactly. His question was, if I've got this structure on the nucleosomes with the DNA, how does it go to the next level of structure? Like, you make this thing that biologists refer to as the chromatin fiber. That's something like, there's a thin fiber, and then there's a thick fiber. The thick fiber is 300 angstroms thick, if I'm remembering right. Was it 300 nanometers? No, 30 nanometers, 300 angstroms. They always say, the 300 angstrom thick fiber. What is this structure? This is one version of the textbook structure, but I believe to this day, this is now more than 40 years later, people are still arguing what's exactly the structure of the chromatin fiber. How do the nucleosomes get packed in there? So that's still, as far as I know, still an open topic, but somehow it's done. And then you make this thick fiber, and then that thick fiber gets folded up in some way, and eventually the whole thing becomes a chromosome with that standard shape that you've seen. And so the live question in 1979 was, what's the structure of the chromatin fiber? So like I was told, go talk to Abe Worcel. Abe Worcel is the expert on chromatin and DNA supercoiling in our, what was then called biochemistry department, later came to be called molecular biology. So he was from Argentina, and he was a very hot guy, hot, temperamental, excitable, could get very animated about all kinds of things. And so I didn't know him, but I walked into his office and I said, I'm in the math department. I have to write a senior thesis. I'm really interested in DNA supercoiling. I hear you know a lot about it. He's like, get to the point. So I said, well, I've heard that there's a good reason to believe DNA is not a double helix. And I gave him this topological argument from Pohl. And he didn't let me get very far. He said, if you write your thesis about that, it will be a laughable matter. I have a very vivid memory of that adjective. He said, that is the stupidest thing. That will be, do not do that. Get out of here. So, and that was the end of that meeting. So I took his advice and didn't work on that problem. But I came back a few months later with some question about elastic properties of DNA. What if it was a twisted structure that had certain elastic moduli, like from continuum mechanics? And I thought it was more respectable, and I worked up. I don't have that much trouble with courage. Anyway, I went back to see him. He wasn't that happy to see me. And I told him, I have a better problem now. Let me show it to you. He said, okay, yes, it's better than the thing you said the last time. But if you want to know a good problem, let me tell you a problem. So then he showed me a certain structure he had, which was, he explained this, what I told you about the 1 3/4 times n versus 1 times n, which at the time was called the linking number paradox. And he said, I have a solution to the linking number paradox. I have a structure that I think would solve the problem, except I don't know how to calculate the linking number because it's a math thing. And he didn't know how to do it. He asked me, do you know how to solve it for my structure? And I said, sure, I could do it this afternoon. And then he said, okay, no, you don't understand. Let me explain it to you again. And he drew a more careful diagram. And I said, no, I really do know how to do it. And I'll tell you tomorrow. So, and the reason I knew, okay, let me tell you what the question was. This is to clarify, what is Abe Worsel's structure, his proposed model? He had taken electron microscope pictures like this, electron micrographs of, this is DNA and the nucleosomes laid flat down on a grid, right? That there's electrostatic forces pulling the structure down and flat. And can you see that there's a very recurrent thing happening in the picture? That very often you'll see two nucleosomes next to each other. Those are I mean, of course, it's very blotchy, not so clear what you're seeing. But the black dots are supposed to be those beads, and you can almost see little strands of what is called spacer DNA between them. It's a little more clear maybe in the next picture. I don't know, are you seeing anything there? Like, can you imagine, whoops, let's see. Like, that's supposed to be a bead, and then there's a little, just a few dots, that's DNA connecting to the next bead, and then more dots connecting to the next bead, and so on. Abe thought that he was seeing this. This is his model. He thinks that's what's happening is that the DNA is winding around these pillbox-shaped nucleosomes in this zigzag structure. It goes from 1 to 2, from 2 to 3, from 3 to 4, over to 5. And it's locally doing 1 3/4 turns around each nucleosome. And the nucleosomes are stacked in this 2-column zigzag pattern. And that's why when it's laid out flat on the grid, you constantly see two next to each other, a dinucleosome structure. He says it's not an accident. It's because that's the native structure and then the electron microscope, which distorts it a little. It doesn't distort it so much that you lose that dinucleosome structure. Okay, so that was his model. But you see the DNA is doing this funny thing where it winds around 1 3/4, zags over to here, 1 3/4, then back. And he asked me, what linking number is predicted by that structure? That was his question. That's what I said. I'll do it have it for you tomorrow. And the reason I knew I could do it was because Francis Crick had written an article saying how you do a calculation like that, which is you go to the store and you buy a piece of ribbon. You buy a piece of ribbon from a dressmaking store or something, and you make the ribbon into the exact shape. And then after you do the thing, you close the ribbon. And then once you've closed it, you've got a topological invariant. You can't change the linking number once it's closed. So what I'm saying there is, like if I take this belt, okay, so this belt is supposed to be DNA. And this is one strand of DNA. Forget that it's not double helical here. You could imagine that it was. Just the thing is, it's the ribbon model of DNA is I've got one strand, I got another strand. And then this is like the equilibrium configuration where everything is twisted the way it wants to be. It looks like it's not twisted as a belt. Then if I start inserting supercoils, so this is supposed to be the axis of the double helix. And then if I make a supercoil, you know, and do something like this, and then close the belt, then, so here I've done one supercoil, and you can see maybe that there's one twist in the structure. And so this strand, if you now trace it, is going to be linked once through the other strand. And the cool thing about a linking number is that it's a topological invariant, meaning if I don't like this twist and I try to get rid of it by untwisting it here, it's just going to be twisted somewhere else. You can't get rid of it, right? It's obviously locked into the structure. Okay, so why that's important is because Crick understood that thoroughly. that I can make Warcell's complicated structure with my ribbon. Then I can throw it on the floor and lay it flat and just count how twisted it is. And I can't change the linking number by doing that. So all I have to do is count, which I did. And so I carefully, it being Princeton, I had some squash rackets available. And so I used those as poles and I wound it carefully in his pattern. And then over on this squash racket's handle. And then on this, actually, truth is, I first did it on the pipes in the radiators in the room, but that was no good because after I closed it, I couldn't take it off the pipes. So that was stupid. It shows you what you're dealing with here, explaining my B-. But after then I realized, no, I need poles, not pipes. So I used my squash rackets in the room and wound it, carefully took it off the two squash rackets, laid it on the floor and counted. And after doing 24 nucleosomes worth, I got a linking number of 24. In other words, Warcell's structure was predicting what was seen in these experiments. And of course, I was very ecstatic and did it a few more times to check that I did it right. And so then the next day, as promised, I went and showed him. And he wanted me to show him a few more times. And after I did it, he started hopping around the room. And he said, this is going to be big. This is going to really be big. This is big. So, yeah, he thought that this was a very big deal. But then he said, but that's just, that's not a proof. Can you give me a proof? You mathematicians are supposed to. So I thought about it for a while, and then I realized, yes, it's actually not that hard to prove. That if you look at his structure of the DNA winding shown in panel B there, how can we prove that that would contribute? Those are, that's a little piece of the structure. What would a proof look like? So here's a proof that if you insert two nucleosomes with this winding pattern, they will contribute -2 to the linking number of whatever you insert it into. In other words, those two are contributing -2. In other words, -1 per nucleus. I didn't mention the negative sign. It creates a left-handed change in the linking number, counts as negative. But so what my proof does is you start with Warcell's structure. You make a double-stranded pass through the structure in part B. Seem to have lost my, oh, here it is. This move where I look right there and then I pass that through. There are actually enzymes that can do that, right? DNA gyrase can do that. There are other, that wasn't known at the time. But there are enzymes that can make double-stranded breaks and pass DNA through itself. And that's what I'm showing here. I've passed it through itself. Notice that's below rather than above. And then once I've done that, I can move this top down around to make this. And the net result of these little transformations is after this double strand break, which changes the linking number by 2, I now have a structure that has mirror symmetry around its, or inversion symmetry around this point in the center. And so that thing, by its symmetry, cannot change the linking number at all. That would do, whatever it does on the left, it's doing the opposite on the right. And so this structure would produce no net change in the linking number by its symmetry, and we got to it by changing the linking number by 2. So anyway, this kind of argument shows you that each nucleosome in Warcell's structure amounts to changing the linking number by 1, which was what was known. And there are some assumptions built into this. I don't want to claim we solved the problem, because he had to assume that there's no additional twisting happening in the spacer DNA between the beads. He was assuming just relaxed connections. So that was his model. Anyway, this was the proof. We wrote, that was my first scientific paper. I've never given a talk on this. It was a fun thing to prepare this talk, because in my now 40-something year career, this is my first time talking about my first paper. I'm not known for this at all. Maybe a lot of you think of me doing only, synchronization and networks and stuff. I never talked about DNA before, so it's fun to have an excuse. Anyway, there it is. We were in PNAS. It was very thrilling. Don Riley was the person who did the electron microscopy. And so in terms of the original question then, what is mathematical biology supposed to be? Here was my first foray into it. And I think you can see this was very much math in the service of biology, right? This is not a brand new theory in math. It's not a deep math question. It's buying a piece of ribbon, doing a little argument that's a proof. But it was helpful. It really was. And I mean, is it big? Was it big? Well, I don't know. I mean, I haven't kept up with this. There are people in the room who probably know a lot about current thinking about chromatin structure. Our model is in the textbooks, but so are other models. People are still arguing. So I don't know if it's going to turn out to be correct or not. I think it's complicated, actually. But anyway, so it was very thrilling. So at this time in my life, I felt this is what I got to do. Apply geometry to biology, or topology to biology. And so part 2 of the talk is about the next attempt to do some of this in connection with phenomena that come up in the study of excitable media, things like nerve tissue or heart muscle. And so this was work that I did with this gentleman, Art Winfrey, who had studied here in Chicago back when there was Jack Cowan's group doing theoretical biology in Chicago. Winfrey, I think, did a postdoc here. I think of him as my main mentor, although Worcel was great too. But anyway, Winfrey had written a book called Geometry of Biological Time. And you can imagine how exciting that was to me, thinking I was inventing this subject of geometric biology. No, people have thought about that. And Winfrey was connecting it not only geometry and biology, but to time, to biological rhythms and things unfolding in time. And so let me give you an example of the kind of thing Winfrey was interested in if you haven't heard about spiral waves or scroll waves. This is a pretty much life or death topic, which is a state of disorder that can occur in a heart where instead of the normal pumping rhythm, you get a disordered electrical state, the lethal kind, called ventricular fibrillation. So I want to show you a video of what that looks like in a medical school class. Let's see here. So what you're going to see is a heart that is twitching, kind of quivering. So the term fibrillation, like the disorganized motion of these heart fibrils, doctors often describe it as if you were to hold a heart that's fibrillating, it feels like a bag of worms. That's the standard metaphor. It's just this jiggly, slippery, nasty bag of worms that's not organized pumping at all. And because there's no good pumping, you're not getting oxygen to your brain or your body, and you'll die within a few minutes if you have fibrillation. So in the experiment, you're going to see the people put on the defibrillator paddles and jolt the heart back into the normal rhythm. Let's see. Oops, I didn't want to do that. Let me go backward. Try again. Is that not running? Hello, try again. There we go. This I found on the internet. That pattern in the EKG is bad. That's what a fibrillating heart looks like. You can see just quivering uselessly. Put on the paddles, clear, bzz, and now you see a normal rhythm. So something is going on that these paddles, these electrical defibrillator paddles, fixed. So what is it that just happened there? Well, you see now it looks like a reasonably normal rhythm after being defibrillated. So people have thought about this for 100 years, and it's known that there's an electrical disorder in a heart that is undergoing fibrillation. And so here are computer simulations that could get from Elizabeth Cherry and Flavio Fenton. They'll give you some idea of how to think about different states of wave propagation, electrical wave conduction in a heart. So let's see here. Right, so if you have a plane wave, just kind of propagating down the heart from the sinoatrial node, the natural pacemaker of the heart, through the AV node and then into the ventricles. Approximately that kind of simple planar propagation of the wave will give you a normal looking rhythm like shown down here. Now, if you happen, instead of a plane wave, have a rotating spiral wave of excitation on the heart, and then there's a real picture from, I guess, voltage, or maybe I don't remember if it's voltage or calcium sensitive dyes, imaging a surface of a real heart, you can see these rotating waves. They've been known to electrophysiology and cardiology for 100 years. The doctors talk about circus movements or reentrant action potentials. In other words, think of like a grass fire that's propagating through a field of grass. It's burning, burning, burning. And by the time it gets back to where it started, the grass has regrown. That thing could just keep going around and around. And so an electrical wave in an excitable medium like heart tissue can just excite the tissue. And then by the time it comes back, if the tissue is no longer refractory, it could just keep doing that. And so something has set up this kind of pattern in a heart. And then the EKG that corresponds to it is nasty looking, like that. Now, that's not fibrillation. That would look like what doctors would call tachycardia. That's a faster heartbeat than you want. You can see the time between the spikes is very much faster, much shorter than in a normal rhythm. So tachycardia is bad, but that wouldn't necessarily kill you. Your heart can still beat, it's just not beating normally. But what does kill you is if this spiral breaks up into lots of little spirals. And so, here's just showing what tachycardia might look like in a 3D simulation of a real heart from Cherry and Fenton. But then if we get this disorganized state where the spiral wave breaks up into lots of little structures, and then you're seeing imaging of that also on a real heart, that will lead to some very freaky looking pattern on the heart like shown there in the simulation. And notice the very low amplitude wave form on the EKG. That's where your heart is not pumping and you'll be dead in just a minute or a few minutes. So the question that has long perplexed people in cardiology is how to describe this series of transitions from normal rhythm to tachycardia to fibrillation. And they figure it has to do with like something creating spiral waves and then causing them to break down into spiral turbulence or something analogous. Okay, so now this is not just something that happens in heart muscle. I mentioned that there's broader mathematical concept of an excitable medium, like I talked about a grass fire. But a classic excitable medium, you don't even have to be alive, like grass. You could just do it in a chemistry experiment with this strange reaction, the BZ reaction that you may have heard of. So in the BZ reaction, you see these blue waves of oxidation propagating through a reduced medium that looks orange. And notice that when two waves bang into each other, collide headlong, they don't pass through each other. When blue meets blue, they annihilate. And that you could think of that as like a grass fire hitting another grass fire, can't burn through the ashes. The ashes are like the refractory zone. So these waves, unlike linear waves, do not superpose. They don't pass through each other like solitons. They collide and annihilate. That's the classic thing in an excitable medium. And you might even see that inside there's little carbon dioxide bubbles produced. Don't worry about those. It's sort of hard to see the spiral waves, but there are spiral waves in the centers of that one growing structure. You could see them more clearly in this picture. If I watch this movie, this is just zeroing in on a little spiral. It's amazing. What's happening here is that you've got this blue zone of oxidation in the chemical reaction, and the wavefront always just propagates normal to itself. It just propagates from excited medium into quiescent medium. It's like the grass fire spreading where it can into unburnt grass. And what's interesting is that with spiral geometry, if every piece of the wavefront moves normal to itself, for a spiral geometry, that makes it look like the spiral is rotating. Sort of like, if you see a barbershop pole and there's that helix on the barbershop, that thing is just rotating, but it looks like it's propagating, right? This is the opposite. It's just propagating, but it looks like it's rotating. And it's a natural aspect of spiral or helical geometry that propagation and rotation are easily mixed up, one for the other. But so anyway, Winfrey himself discovered spiral waves in the BZ reaction in 1972. And he was very interested in their possible connection to cardiology, since people had known about these rotating action potentials in that setting for a long time. But what he was especially interested in was what is the three-dimensional generalization of spiral waves? Because the heart is not just a surface. The heart is a big, thick piece of muscle, especially the ventricle is very three-dimensional. And so Winfrey figured that these spiral waves that you can see on the surface must actually penetrate into the muscle and make something that's not just a spiral. What would it be? He called it a scroll wave. Sorry, the picture would be like this. You have a spiral cross section. Like in math terms, a spiral across an interval is a scroll, right? So a line of spirals would be a scroll wave, like a scroll of paper. And I want to just go back. I'm a little out of order here. At the center of this spiral or scroll is what I'm going to call a singularity. And so let's talk about what's singular about it. So as I say, when you have a rotating spiral wave, if you think about, just fix your attention on one point in the domain, that point will go through a cycle of I'm excited after the wave passes through me, and now the wave is rotating around, and now I'm getting refractory because I was just excited, and now I'm gradually getting less refractory, and now I'm quiescent, and I get excited again the next time the wave goes through. So there's a whole cycle of excitation at any point, and so you can define a phase at any point by thinking about this cycle. Like we could say, as Winfrey wrote in Geometry of Biological Time, he means 1 and 0, not that 1 equals 0, but that 1 and 0 correspond to the same phase. They're both, let's say, the excited phase, where in the picture, the spiral wave happens to be right there at that time. So that's a place of excitation. 1 is its phase, or 0. And then all of these points on the solid line are points of equal phase. Now, a tenth of a cycle later, that wave will have propagated to here on the dashed line. And so all these dashed points are at phase 1/10, right? A tenth of a cycle later. And then a tenth of a cycle before, 9/10 of a cycle would be 1/10 before the end. That would be this dashed line. So the thing is, you can then choose, you could basically define phase everywhere in the medium, except for at the one point at the center where all the different isophase contours meet. So at that point, it's like saying, what's the longitude at the North Pole? Or what's the angle at the center of polar coordinates? It doesn't make sense. There's a phase singularity at a place like that. And so that's what I mean when I speak of singularity, that you can't define phase there. And it's interesting because it seems like it's sort of it's the point around which the spiral rotates. It's an important place. Okay, now, if that's phase, as I say, it's not defined what the phase is at the center. You've got a phase singularity. So when you extend these ideas to 3D, you've now, instead of having a point of singularity, you have a filament of singularity. Okay. So Winfrey's idea was, I want to figure out the structure of scroll waves in thick, three-dimensional, excitable media. Because Winfrey, think this is related to what's causing fibrillation. If I can figure out how 3D waves behave in thick heart muscle, or maybe I do simulations in 3D BZ reaction, maybe I'll learn something about this 100-year mystery of fibrillation. So he set out to think about what are all the possible ways you could have 3D scroll rings. And it's rings because he figures the filament is not just going to have like dangling ends. You don't expect that. You'd expect the filament to close into some, I mean, it could hit the boundary of the medium. But if it doesn't hit the boundary, you don't have phase singularity just hanging there like an electric, you know, like a downed power cable. It's going to close. So, but then he realized there's a topological issue because there's a lot of ways of closing a scroll ring. You could do this picture. You could take a spiral wave and just go around with no twists in it and just close it simply to make an untwisted scroll ring. But you could, you know, if you like geometry and topology like Winfrey did, and here I am, it's now 1981, and I'm reading his book, having thought about DNA and linking numbers, and I'm reading about this thing with heart rhythms and geometry again. He said, well, couldn't it also do this? Couldn't you put a full twist and then close it? Is that possible? Could you have a twisted scroll ring? Is there any constraint on how many twists you could put in? Could you tie knots in it? Could you have different scroll rings linked through each other? What's possible? Is everything possible, or are there rules? And so he posed that question in his book and said he needed help with it. And I thought, man, this is the most exciting thing I've ever read. So I asked, could I come work with you? And he wrote back in characteristic, very strange fashion and said, population density is identically equal to 1 in my lab, which means he never had students. Nobody ever wanted to work with him. He was very weird, and he was in a biology department at Purdue. His background was engineering physics. He loved doing topology. And as I told you, he discovered spiral waves in chemistry. He was impossible to classify. He was really strange and also very blunt in a, what we now, I guess, would recognize as being on the spectrum kind of way. And that's not an official diagnosis, but it's obvious to me that that's what was going on with him. And so anyway, after I wrote to him, he wrote back 5 minutes later and even said in the top of the letter, 5 minutes after receiving yours, impulsively, Art Winfrey, yes, come to work with me. The population density identically 1. I would love to work with you. And I had told him I had thought about linking numbers and twists, and I wanted to help with the twisted scroll ring problem. So the first thing we started to do was work out some of the math. Now, he had done this part in his book. He realized that if you had a twisted scroll ring, then it couldn't be there by itself. That would be impossible. So what he's showing in this picture is he imagines a twisted scroll ring is sitting inside this torus. Like imagine a glass torus surrounding a scroll ring with 1 full twist. And he's going to measure in his mind what is the phase at each point along this interior disk that sort of plugs the hole of this glass torus. So like he supposes, because of the twist, if this is at phase 0, the twist is going to cause these other points to be at different phases. And it's going to make, if you had 1 full twist of the scroll ring inside, you're going to have 1 full winding number, a winding number of 1 as you go around this disk. The phase will advance by 1 full cycle around that disk. I don't know if you can see that. But he could see that in his mind's eye. And he realized that now if I want to take away the glass torus, basically he wants to extend this twisted scroll ring to fill a 3-dimensional medium like a heart muscle. And he wonders, can I extend this structure outside of its, the glass torus is just for your mind. It's just a boundary that's not real. It's just to help you think about confining this complex structure. He wants to remove that thing and extend it. Can I smoothly extend the structure to fill in this disk? And so what's the problem? The problem is if you have a disk with phase defined everywhere on it, but it has a winding number of 1 on its boundary, then think of it this way. Winding number meaning, as I go around the boundary once, I go through a full cycle of phase. Now imagine I go around a slightly interior circle to that. I can't draw it so well. Draw a smaller circle. What's the winding number going to be on that smaller circle? It's still going to be 1, because as you continuously deform the circle, you can't change an integer continuously. It has to stay constant, right? Integers don't, right? So the winding number is going to be 1 all the way down till you have just a point. And so this is what it would look like. You'd fill in the whole disk, and there would be a phase singularity inside it. And you can prove that with topology, with this little result. So that tells him that somewhere inside this imaginary, somewhere inside this hole, there's another phase singularity, which means there's another filament linking through. You can't have a single twisted scroll ring. They have to be in pairs, and they have to be linked through each other. So that was an example of the kind of rule he was looking for, and he wanted to know the rules in general. And so first what we did was make improved graphics of what he had drawn by hand. This got us on the cover of Nature. That was very, very thrilling. But in 1982, so after working with him in West Lafayette in the first summer, he was never my advisor or anything. I just went to work with him in the summer. And actually, for the first 2 months of that first summer, he had me doing BZ chemical experiments. Now, remember what I told you about when I did chemistry? So I sucked at that still. And finally, I said, I think I should be doing math about this kind of. And then he said, OK, help me figure out the topology. And so we came up with a lot of nice topological rules about what was possible. And then the second summer, he said, I want to make pictures and really see this. And so we went to Los Alamos, where they had a Cray supercomputer, which was very exciting at the time, to work on what they called the X machine. And so we made these graphics there with the help of some of their graphics gurus. That's what an untwisted scroll ring looks like on the top. The twisted one is kind of pretty. And then we tried to extend it by letting the lip of this this little thing just keep going and going, and ask what happens. Now remember, the surface can't pass through itself, because when a scroll wave or a spiral wave hits itself, it annihilates. So we can extend it as far as it can go till it hits itself. And so this is what it ends up looking like. This one, you can probably kind of imagine an untwisted scroll ring. This doesn't have any real problems. But this twisted one is really gnarly down the middle. And in fact, what you're seeing is a new kind of phase singularity predicted going through that the topological argument told you had to be there. But now with computer graphics, you can really see it. And so in the course of that paper, I mean, we wrote a series of papers, but that one was a review. This was a little principle that we came up with, which we thought of as kind of like the Pauli exclusion principle for, can you make hydrogen or helium? What are the rules? We had an exclusion principle for what topologies of configurations of scroll rings could work, would not violate any chemical principles. And it was framed in terms of linking numbers. In fact, it was very convenient that I already knew something about them. So I won't go through the details of it, but the point was there was a kind of quantized periodic, not periodic really, but I mean, there was a table of possible structures that were predicted, and they're still predicted. And okay, now what does it have to do with cardiac arrhythmias, with fibrillation? We don't know still. It's now a long time since then. That work was in the early 80s. It's very hard to observe these. People have observed an untwisted scroll ring in the ventricle. That's what this paper shows. So this is a 2018 paper. And I've just highlighted the key point from the abstract, that three-dimensional scroll waves and filament-like singularities can be observed deep inside the heart wall. But that's fairly recent. And that was in our classification, the untwisted scroll ring. To set up the initial conditions to get these other knotted or linked structures is very difficult. You can do it in supercomputer simulations, but they may not even turn out to be relevant to actual arrhythmias. That's just the way nature turned. I mean, nature isn't using this topology as far as we know, or it's very unlikely to happen, I think. So what's the point then in terms of our question of what is mathematical biology? To me, this was kind of a two-way street where the biology suggested some really interesting math problems about topology and excitable media that it was a pleasure to work on and figure out. And maybe we gave some insight into some biological problems, but I don't think a whole lot. And it turned out nature didn't cooperate, or as far as we know, at least in this example. All right. So I guess the question is, should I follow the basic rule of lecturing, which is never go over time? I could, in which case I could say thanks for your attention, and we could be done, and we could take questions. That's the plan, right? At 6 o'clock, we start questions. I did have a third story for you, but I realize I've been, very slow, but I don't want to rush. I want you to understand the question. So I'm okay with stopping here. I have no particular agenda. If you're dying to hear more, I could do more, but it would take another 15. It will take 15 minutes more. Okay. Well, you don't have to do it seriously. All right, I'll give you 15 minutes. I promise I'll stop at 6:15. Okay. All right. That was nice. Thank you. Sorry it's taking so long. All right, so here's the last one. This is a topic that is, very near and dear to my more grown-up heart. Self-synchronizing systems, mostly done over the years with my friend Rennie Marolo. And so one of the papers we worked on was about, people think of it as a paper about fireflies getting in sync. And the thing about fireflies, or even the heart example that we just talked about, is that a lot of times in biology, these oscillators are not like the oscillators you learned about in freshman physics, right? Freshman physics, you've got a simple harmonic oscillator, a mass on a spring, bobbing up and down. In biology, there's often jolts. There's like an action potential, zap, a spike, or there's a firefly that is dark in the dark and then suddenly flash. In other words, there's pulsatile behavior. And so I want to tell you a little story about self-synchronizing systems that communicate with jolts or pulses. It makes the math much harder, but it's more biological in many cases. And so first, to illustrate what I mean by self-synchronization before I talk about biology, let me show you an inanimate system that self-synchronizes. If you have never seen this, you'll be amazed that this is even possible. These are just metronomes of the type that musicians use to keep time. What you see in the video is 5 metronomes. They're on a little board, and the board is going to be placed on top of those 2 empty soda cans that are going to be used as rollers. But before we do that, I just want to show you what happens if you don't place the whole apparatus on the rollers. Now let's see, here I may need to put up the sound. Here we go, volume. Okay, so first, you're just going to start the metronomes ticking, and you won't hear or see anything dramatic because they're not interacting strongly enough to get in sync. Then the gentleman will place them on the rollers, and that will, as they tick, they're going to cause the platform to move a little bit. And that will put all the metronomes in this non-inertial frame that puts forces, effectively fictitious forces on them, that's going to jiggle the metronomes, and they're going to jiggle themselves into sync, almost like entropy going backwards. Except, well, it's an open system in the sense that it's expending energy. It's not really a close. Is it a close? The thermodynamics is confusing, but this is not defying. It doesn't defy any laws of nature. You've pumped up energy by winding up the springs in the metronomes, right? And eventually they'll all stop ticking. So you will get to equilibrium. There's no violation of the second law, don't worry. There's not much that's going to happen. They're done. Okay, I'm not going to let that go. So in other words, they did spontaneously synchronize. They're not perfect. I mean, you may have heard that they're a little bit off even at the end, but they're very close to being in sync. Now they're not, because don't have exactly the same natural frequency and whatever. There's always imperfection. Just like in biology, there would always be some diversity in natural frequencies of any real biological oscillators. But still, they somehow, through these mechanical forces, got themselves in sync. Now, it's not just a frivolous thing. Here's a case where human beings did the same thing. Opening day of the Millennium Bridge. I'm sure some of you have walked across the beautiful Millennium Bridge in London, footbridge across the Thames. On June 10th of the new millennium, the Queen was there, the BBC was there filming, 80,000 people from London showed up. The police said, okay, everyone can go on this beautiful new little thin, ribbon-like bridge across the Thames. And within a few minutes, something weird started to happen that the engineers didn't know about and nobody anticipated, which was that the bridge started to move sideways. Now, you may have heard this old thing about that soldiers marching in step across a bridge are ordered to break step. And that's true. There have been incidents where soldiers, with the natural frequency of human walking, which is about 2 cycles a second, if the bridge happens to have a vertical resonance frequency of 2 cycles a second, you can induce resonance in a bridge and cause it to have big vertical oscillations that can, and have in some cases, cause the bridge to break and the soldiers to drop into the river, and it's bad. So you don't want that to happen. And every civil engineer knows you don't build a bridge with a vertical mode of 2 cycles a second. This is not what happened on the Millennium Bridge. This did not move up and down. This moved sideways. Okay, it's wobbling. It was referred to as the wobbly bridge. And why is it moving sideways? People don't put big sideways forces on a bridge when they walk on it. They put vertical forces, but sideways? And also, you'll see in this footage from BBC, from a show called Science Shack, that the people are walking in step. And it's kind of like this. The bridge is moving sideways for whatever reason. And that's uncomfortable. If you've been on a train that's going fast and it starts jiggling, you'll put your feet out wide like a penguin and you'll start walking like this, usually with the same frequency as the motion. It just feels more comfortable. Except that on a flexible structure like the Millennium Bridge, as more people start doing this, they start to drive the bridge, which makes it move more, which makes more people go like that. And that's what you're going to see.
The most dramatic and shocking footage shows whole sections of the crowd, hundreds of people, apparently rocking from side to side in unison, not only with each other, but with the bridge. This synchronized movement seemed to be driving the bridge. But how could the crowd become synchronized?
Yeah, so it's a whole long story, to think about the interaction of people on an elastic structure. It's complicated, and people are still discussing what really happened on the Millennium Bridge that day. But I hope, you recognize that this study of self-synchronization could be pretty impactful. Nothing bad happened, by the way. The bridge didn't collapse. It's still there. Nobody got hurt. They closed the bridge. They spent about close to 20 million pounds to repair it. They put in big shock absorbers underneath, which you can see next time you're in London. So they made the damping much larger, and the bridge doesn't do this anymore, which in a way is disappointing. But it's considered safer, and so, okay. Right. So, oops, where did I go? Yeah, so lots of living things self-synchronize, not just pedestrians on the bridge. I think here you can see a little movie of some neurons flashing. They've been treated with a dye so that they light up when they fire. And so it's not so unusual. The fireflies I mentioned, sometimes male fireflies will gather in trees and all flash on and off in unison in places like Thailand or Malaysia. So it's a widespread phenomenon in biology. And I just want to finish then with this quick analysis of a model from Charlie Peskin at NYU, a really simplified model of what's going on in a situation like this. So you could say, why would Peskin or anyone do such simple models? And it's because the problem is really quite hard. to do properly. First of all, the individual oscillators, as I say, they're not linear oscillators like simple harmonic oscillators. They won't synchronize if they're linear. They have to be nonlinear. So you're not going to solve them exactly. There's always, in real cases, 2 or more timescales, like a fast timescale of the spiking and a slower timescale of phase adjustments to getting into sync, and sometimes more than that. You've got these non-smooth behaviors of spiking, impulsive coupling. If you were really describing a heart cell, let's suppose we're doing the pacemaker cells in the heart, could be 100 ion channels to be thinking about, and also many oscillators. Could be 10,000 cells in the SA node of the heart. And also, it's not just connected in a simple way. There could be interesting geometry or topology in the graph theory part of the problem. So there's a lot going on in the study of coupled oscillators. And so when Peskin made this model of the sinoatrial node back in 1975, he just thought of it as sort of like, what's the simplest problem that is still difficult, that could maybe contain the essence? And so he thought about relaxation oscillators, which he thought of kind of like as in electrical engineering, if I'm charging up an RC circuit. So a capacitor is kind of like a cell, because a cell has a membrane, there's an inside and an outside. And as you're charging up, and maybe it's leaky, that's why there's an R in the RC circuit. So you're charging up until you reach a threshold of voltage, and then you fire, then you instantly go back to 0, and then you charge up again till you hit the threshold. That's his model of a individual pacemaker cell. And for simplicity, suppose they're all identical, and they just all obey this little differential equation for an RC circuit. And his rule is when a cell reaches a voltage of 1, that's the threshold, it fires, and then instantly jumps back to 0, the baseline. And another cell that receives that impulse gets kicked up by some amount that Peskin called epsilon, which he thought of as a small number. And if in being kicked up by epsilon, that cell gets kicked over threshold itself, then it can fire. Otherwise, it just gets kicked up by epsilon and continues charging up from there. So that's his model. And he conjectured that with that model, this system would always self-synchronize. As he put it, it would, for arbitrary initial conditions, all the oscillators would end up firing synchronously. And I read about this in Winfrey's Geometry of Biological Time. And Winfrey mentioned this, a nice question about pulse-coupled oscillators, not because Peskin could only prove it for 2 oscillators. He did prove that it would work, but with 2. And the population problem for an arbitrary number was unsolved. I mean, that Winfrey's book was a goldmine. It had so many open problems in it. And so I ran a little computer simulation of Peskin's model with 100 oscillators, where what I'm showing here is if you divide time, a natural period, into 10 bins and count how many of the oscillators are firing in each of those 10 bins, at first it's like bop, 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 bop. They're firing all over the place. But then gradually one peak starts to emerge, and now all 100 are firing bang in that bin, and then bang again. So it did self-synchronize fairly quickly, as Peskin conjectured it would. Excuse me. So it seemed like his conjecture was true. Sorry. And he also said that there was a robustness to it, that it would still work even if the oscillators are not quite identical. So Rennie, my friend Rennie Merola, is a really good mathematician, and I asked him to think about this with me. So we looked at Peskin's proof for 2, which he did for, like a perturbation theory calculation. He assumed a small kick, epsilon, and small curvature. This gamma controlled how concave the shape was. If gamma was 0, you just go up on a straight line. And he found that he needed both of these effects. He did need concavity. If you think the result is sort of obvious, that identical oscillators kicking each other, of course they'll get in sync, that's wrong intuition. If they rose up to threshold, if they accelerated up, concave up, they wouldn't get in sync. With 2, they would get into antiphase, turns out. So something about the curvature matters as well. But it wasn't very clear from Peskin's perturbative calculation. So what Rennie and I did was prove this conjecture for any number of oscillators, not just two, but any arbitrary number. And we didn't need his Peskin's RC circuit dynamics. The result was generically true as long as the curve, this charging curve, is increasing and concave down. It was only the geometry that mattered. And so I won't go through the details of that proof, but we just looked at some curve with the desired shape. Using it, you could calculate. Let me give you the idea of the strategy. It's in dynamical systems, we would call it calculating the Poincare map. So that what it means is, let me suppose one oscillator has fired and I have n-1 other oscillators that are somewhere. And then I wait till the next time that given oscillator fires again. And then I look at where those n-1 other guys are the next time. And so I have a map from this n-1 set of phases to itself, and I want to understand something about that map. And so here I'm calculating the details of that map for the case of 2, but the generic idea of the strategy, as I say, is I'm going to do the Poincare map. And the main thing that it needs to be shown is that this map expands volume. So you have this n-1 dimensional space. You can calculate its volume. Basically, you have to look at a determinant. In multivariable calculus, we teach that the determinant of the Jacobian tells us how volumes or areas change from before and after a transformation. It's the factor. And so if the determinant of the Jacobian is greater than 1 in magnitude, you're expanding volume. A region gets bigger in volume under the mapping. And why that's important is, and that's where we use the concavity and the upward kick and all that, and the monotone increasing. All that comes into the proof that it's a volume expanding map. Why is that important? Because if you think about the set of bad initial conditions, meaning the set of initial conditions that would not lead to synchrony, that set, by definition, these are the points that will not lead to synchrony. After one iteration of the return map, they still won't lead to synchrony. So that set gets mapped into itself. If you're bad after one iterate. So the bad set is mapped into itself, but its volume got bigger. And you can't map something into itself if it got bigger unless it had 0 volume to begin with. And so with that kind of argument, we show that the bad set has measure 0, which means, although it's not empty, you have no chance of hitting it. And so that proved Peskin's result. And so I want to conclude with this point, which is, why study a simple model like this, given that a real heart cell or a firefly is nothing like this? It's because it's so obviously bad that it's easy to think of how to do better, right? It invites improvement. Any sensible person reading our paper thinks, I could do better than that. Why didn't they do blah, blah, blah? And then that person cites you. So like Bill mentioned all those citations. That may be a lot of, like 50,000 critical citations. So you have to look at what the people say when they cite you. It might be, this paper is the worst thing ever. Hopefully not all 50,000. This paper didn't get 50,000, but it did get a lot of citations, and it was a lot of them finding ways to improve or generalize the model, and also to suggest spin-offs into other fields. So recently, I mean, here's an example just from the recent literature. People in their own disciplines know of a lot of things that are like, something is charging up and then it fires and then it resets. It's come up in neurons in thinking about cortical networks, but the electrical engineers thinking about wireless sensor networks have used this kind of basically firefly type model to get self-synchronizing systems of clocks. Or even the social psychologists thinking about why people synchronize, like interpersonal dynamics between a psychiatrist and their patient. There are measurements about people's heart rates and stuff. That interpersonal synchrony literature is drawing on our model. Or recently, Itai Cohen and collaborators did some work on microscopic machines. We're talking about micron-sized little robots that are coordinating in swarms, and they're getting themselves in sync as a collective to coordinate their timing using the pulse coupling strategy that we, again, it's not what biology is exactly using, but it works fine for these little robots. And it's even being used in machine learning these days, where instead of just doing gradient descent in the usual way, just down to a minimum, people are thinking about it in connection with neural networks that aren't the usual neural networks that only have fixed points, but neural networks that can spike in the machine learning sense. So that's part of the fun with simple models, that they can be very transcendent. They could be useful in many fields. All right, so thanks for all the time you've given me. That being the third case where bio inspired math and many other fields. And so as far as my own question, I mean, I'd like to hear your answer, but I sort of think let a million flowers bloom when it comes to math and biology. There's a lot of ways to do it. Okay, thank you. No synchronous applause. Now that was a perfect opportunity. Okay.
All right, we do have time for some questions if anybody in the audience has a question.
I give this a more philosophical question. So kind of related to what you started, like there's this whole question, mathematics, is it an invention or discovery?
The question was, it's an old philosophical question, is math invented or discovered? And you know, the feeling of a mathematician generally is when we do our work, it feels like we're discovering something, that it's out there, that these are properties that existed whether we thought about them or not. I mean, Archimedes has a beautiful quotation about that he says these certain properties are inherent in the figures all along, and he was just lucky enough to understand how to calculate the volume of the sphere. Like that was there before he discovered it. So most mathematicians in practice believe that the ideas are being discovered, but that's a really philosophically problematic thing, because where are those ideas if they're not in human beings' heads? I mean, we say they're out there. Where? What are we talking about? So I think it's easier to defend the argument that math is invented, but it doesn't feel like it. And in my heart of hearts, I feel like it's discovered. The crisp answer would be that we invent the concepts, but we discover their consequences. I think that's the truth. We may invent the idea of a circle or a sphere, because there aren't really perfect spheres in nature or perfect circles. So we invent idealizations, but once we have them, there are consequences if you accept logic. And so I think those are being discovered. I don't know, do you have an opinion, or is it what do you think? You're a Platonic person so you think that it's often associated with Plato, this idea that there are ideal forms or things, that there's a realm of reality, which is what, those are like the shadows on the wall of the cave in the old Platonic parable, right? That we're prisoners, we're chained, we're looking at a wall in a dark cave, and we see flickering things going by on the wall, which is there's stuff happening out there in the world, except it's not really the real world. It's this Platonic realm, and we're seeing projections of that perfect world onto the wall of the cave. That's all we can see. And if we could turn around and see what is really real in the Platonic realm, the light would be too bright and we'd be blinded, right? So you can't really see the ideal forms except in your mind. And Plato says that's why you have to study geometry. It's an interesting argument that if you want to really understand what is true, you must study geometry, because it's warm-up for thinking about the ideal, as opposed to dirty reality, which is just shadows on the wall. I love that idea, but come on, you got to admit that's pretty murky stuff. Like what, really? I don't know. Because we know that people exist and they have ideas and invent things. I don't know. I want to believe it. I'm with you, but I find it hard to defend. So the question was, could I run a simulation backward if everything is spiking in sync? Was that the question? Start with synchrony and now runtime backwards, what would we see? Well, so in a lot of systems where if the synchrony was stable so that it was being attracted into that state, then when you ran it backwards, yes, things would fall apart. I mean, I think would typically be what would happen. So it's attracting in forward time, but it will be repelling or unstable in backward time. Does that seem bad to you? Yeah. Oh, in the proof, you're interested in that proof, maybe? Yeah, no, in our model, if we would run it backwards, they would fall out of sync. Little differences would get amplified. So it wouldn't resynchronize in backward time. I mean, at first we were a little surprised in our proof that we were using the expansion property of the map. Your common sense might have been the map should be contracting, and I'm going to use the contraction mapping theorem to prove that there's a stable fixed point and that's why it gets in sync. But that's not actually the way the argument works. Luis had a question, or someone else in the back. Yes, please. I missed the beginning of the question. How is all the time I've spent trying to educate the public? Oh, how has it helped me as a researcher to be interested in public communication? Well, it certainly is helpful to be generally interested in communication. I don't see it as so much about the public. To me, it's about the whole job. I mean, whether it's teaching a class or working with a collaborator, or writing a grant proposal, if people can understand what you're thinking and maybe get excited about it, it's certainly professionally helpful, because so few of us do that, right? I mean, so much, think of how many papers or seminars you've had read or attended, and how often it's impossible to understand what's going on after the first minute or two. Or after reading the first paragraph, like the abstract, you could maybe get through the first sentence or two, unless it's really in your field. So I feel like it's a big tragedy that so little effort is devoted to improving our communication, especially in math. I don't want to speak for biologists, although when I look at the biology papers in Science Magazine or Cell, I think, come on, I studied some biology. I should be able to read this. And it's so hard. So I understand why, but I think that's partly helped by certain things on the internet now, like when people write threads or micro blogs about their new paper, those are often really fun to read. Like you can actually learn what the paper is about by reading someone's account on X or Twitter or Instagram. Anyway, so, but no, I don't, am I answering the question? Was that what you wanted? Yeah. No, I think we all have to really work hard on trying to, I mentioned at lunch with the students today that it's not enough to be clear. Clear is important, but it'd be really good to help people love the question you're thinking about. Because so often, even a good expositor will give a clear presentation, but you don't know why you should care. And I went super slow. I'm sorry it took so long, but I wanted you to love the different problems. I think that's what I hope you'll come away with is think about the questions and how, like think about your own work. What do you want people to know? Don't rush into the answer. Make them love the question first. That'd be my bit of advice if I could leave you with one piece of advice.
All right. That's a good note. Let me,
Okay.
before you thank Steve again, let me just say there's a reception for people that use the Eventbrite to register for this talk. The security people, it's behind a security desk, which is a little bit complicated, but they have all your names so you can get past security. Anybody that has a Northwestern ID can go past security. The reception is just across the street. It's the modern atrium in the medical school. There's a door across the street that says Feinberg. You just go in there and go through security and you can get to the reception. Okay, and With that, please thank Steve again.